If you're at home or in here and you want to try to go ahead and log in, he's on a computer that did not save his password, so he's got to look it up. But I will go ahead and introduce Anthony Christidis, who will be giving a package demo today on scDiagnostics, diagnostics functions to assess the quality of cell type annotations in single-cell RNA-seq data. And he's got to get his password. So let me go back to here. Or I don't know if you want to kind of, it's good teaching to kind of do it as people do it.
Ah, good. Yeah, so just in case you want to follow on Galaxy, it's the scDiagnostics workshop. I also have a link to the Docker image on my repository if you want to run it on your own without using Galaxy. But all right. And then when you open the workshop, you will want to go to vignettes, and then there's only one vignette, and that's the one you'll have to open. All right. Is everything set up? I can start with the screen is shared and everything is fine? Okay, thank you. Okay, yeah, so so my talk is scDiagnostics. I'll be talking about diagnostic functions to assess the quality of cell type annotations in single-cell RNA-seq data. I'll describe more what I mean by that in just a second, but first I'll give a couple of acknowledgements. So yeah, I'm Anthony Christidis. I'm a comp scientist at CCB at HMS. And then the work I'll be presenting today is joint work with many great people, including Andrew Ghazi. He's actually online. He will be answering questions for the coding portion of this talk. He's a statistical geneticist. And then there was a postdoc, Smithu Chawla. Nitesh Turaga was a consultant. And then obviously Ludwig and Robert, who was, they were instrumental for the development of this package. Okay, so why is the topic of developing diagnostic functions for cell annotation so important? Well, if we just take the workflow for cell type annotation, there's really two approaches we can use. So the first one is, as you probably all know, marker gene based annotations. So the typical workflow for this is to just do cell clustering, and then you do some differential expression to find genes that are differentially expressed for each cluster. And then we can look up some marker genes for different cell types. And finally, we can assign the label for that cluster based on the marker genes, the differentially expressed genes. And then the second approach for cell type annotation is what's known as annotation transfer, which is we typically have a reference data set, and then we have a query data set. And for example, there's the package scR that uses some function that's based on Spearman correlation and nearest neighbors to assess the cell type of the cells in the query data set based on the reference. So it's kind of a way to do automated cell annotation. So these are the two main ways to do cell type annotation right now in the typical workflow. So what can go wrong? So for example, if we look at the marker-based approach, the RNA levels might not be, there might be a discordance between the protein and RNA levels. So for example, if you're looking at marker genes, you might get some erroneous results, right? In an ideal scenario, you would get what you have here in the middle, which is a bimodal distribution for the other cell types, and then you have the specific cell type. And this is a clear indication that you have a marker gene, right? But this is an ideal scenario, and it's not always the case. Something else that can happen is if you have a condition, right? So if you have cells under different conditions, the RNA expression might differ. And then if we look at reference-based annotation or annotation transfer, one thing that can go wrong, for example, is our reference data set and the query data set don't even have the same cell types, right? But then if we use annotation methods like single R, it's still going to give us a cell type for the query data set, right? And it might give us a confidence level, but it would still give us a cell type. In an ideal scenario, the query data set and the reference data set are well aligned, and they have the same cell types, but obviously that's not always the case. And then in another example, we could have a reference data set with 2 cell types. The query data set also has those 2 cell types, but it has an additional cell type that is unique to the query data set. And again, what would happen in this case is these cells would be assigned a cell type based on the 2 possible cell types in the reference data. And it wouldn't really tell us whether this 3rd cell type actually aligns with the existing 2 cell types in the reference data set. So our work is not the first attempt to develop diagnostics for cell type annotation. There is a chapter in the single R book. If you go through the slides, which are on the GitHub repository, it links to that chapter. It's actually a very short chapter because there's not much that's been done for diagnostic functions. As an example, they have a method here that gives sort of a confidence score or an annotation score for cell types, right? But there's really 2 problems with that. Is first, again, if some cell types are unique to the query data set, you'll still get an annotation and an annotation score. And then in the case that you have an ambiguous cell type annotation or a low score, it doesn't tell you why, right? You can't really visualize whether it's in a reduced dimensional space, why the cell type might differ from the existing cell types in your reference data, right? So the goal here is to develop statistical methods that can tell you whether things are well aligned and you have mislabeled cells in your query data set. So when we talk about a reference data set, just for context, we mean a well-curated, expert annotated data set. And then, again, I put sometimes here because, so ideally cells have been labeled by domain experts using known marker genes and experimental validation. But obviously, this is an ideal scenario. It's not always the case. So just because we say a data set is a reference data set doesn't mean that it's perfect either. But it should serve as a ground truth for cell type annotation transfer. And it can be used to train models, identify cell types, serve as benchmark data for new annotation methods. One such data package in Bioconductor that has a vast collection of good reference data sets would be in the CellDex package. For example, I think everybody here is familiar with the human cell atlas, the mouse cell atlas, or a bunch of other data sets in this package. Yeah, any questions so far? Everything's fine. Okay. And then for the query data, this is the new single cell RNA-seq data that needs to be analyzed and annotated. Oh, yeah, sorry. I can hear you. Yeah.
Online. Okay, I'll just, okay, great, yeah. Could you go back to slide 4?
Yep.
Yeah, so here in the marker gene expression framework, could one argue that, you know, on the right-hand side, that plot where you have a cell type where you expect, let's say in maybe let's say context 2 is the expected behavior, and you're seeing some cells of that type in context 1, or maybe by context we could mean maybe different regions spatially, close to tumor, far away from tumor.
Sure.
Could you argue that that's actually an interesting thing, and it's not something to be diagnosed, but it's something to just be looked at like it's biologically interesting? I could imagine that sort of thing happening because of artifacts, which is where scDiagnostics, like this thing it's like a pathological thing, and you want to remove it, or like, but it could also be useful or biologically interesting. I mean.
Yeah, I totally agree. And in fact, most of the diagnostics in the function would identify such a case. When we say diagnostics, we don't mean to completely, we don't treat it as an outlier or an anomaly and we remove it from the data set. It's more to see where it aligns based on the reference data set. That's what we mean. So it doesn't mean that we're going to remove it at all. Yeah.
Thanks.
Yeah, thanks. Thanks for the question. Okay, so for the query data set, this is the new single cell RNA-seq data that needs to be analyzed or annotated. The starting point for scDiagnostics is the query data already has been annotated by some methods. And then our goal is to check whether the alignment is good and the cell type annotation is good as well. And there's a bunch of visualization functions, things. We're going to use a lot of projections to lower dimensional spaces to visualize things, to see where things fall. And yeah, the goal is to check data set alignments to ensure that the query data aligns with the reference data. And we're going to check whether the cell type annotations seem to be accurate. There's also a bunch of functions I've implemented in the package that can also identify anomalous cells, but it doesn't mean that we remove them. It just means that they deviate from what you would expect based on its assigned cell type. Okay, so the core functionality of scDiagnostics is, as I mentioned, dataset alignment checking. There's a bunch of functions I'll go through for that during the coding portion. Annotation validation, and some example functions that were developed for data alignment checking or PCA subspace comparisons, canonical correlation analyses. I'll do a bit of something called optimal transport theory with Wasserstein distances. I won't get into the math of it, but yep. And then for annotation validation, there's going to be stuff like discriminant space projections, PCA projections, and then distance and correlation-based analyses. I'll go over hopefully most of them, but the vignette is very complete in that if I don't have time today, you can go over it on your own, and hopefully it'll be clear. And if there's any questions, you can give an issue on GitHub, and I'm more than happy to answer your question. And then, yeah, there's going to be anomalous cell detection functions. The main working engine right now in the package is going through something called an isolation forest. But I'm not entirely satisfied with the current way to do it. And there's a lot of, so my background is purely statistics. And then there's a very hot topic right now in robust statistics, which is the direction of outlierness in high dimensional statistics. And it's a way to, in a high dimensional space, identify what we call correlation outliers. So these are outliers that are not marginal, but in a high dimensional space, they're hard to detect. But there's methods now to actually detect them. So this is more future work. And yep. Okay, so just a quick overview of what the workflow looks like for scDiagnostics. You have a query data set, you have your reference data set, and then there's going to be a bunch of different diagnostics functions. For example, where we can compute something called the distance matrix. And then there's going to be a bunch of visualization tools to check whether things align well or the annotations seem to be correct. Okay. Just to give a motivation example, this is a real data set with a real result. So this is from the Organ Atlas data from the scRNA-seq package. The data has gone through the usual pipeline from the OSCA book, if you're familiar with it, QC, normalization. And then what we'll do is we'll split the data. 70% will be treated as a reference, and then 30% will be treated as query. Now, what's interesting about this data is we actually have the expert annotation for both the reference and the query. It's part of the data. But what we're going to do is do annotation transfer from the reference to the query using single R. And these are the results. And as you can see, there's a lot of potential issues based on the annotation from single R. In particular, if you look at, well, there's really two that really pop out. So the first one is there's a bunch of, according to the expert annotation, CD8s that have been annotated as CD4 by single R. And then also the myeloids are kind of all over the place, right? So we'll see, and I'll show it just now, again, as a motivation, one particular tool that was developed for the package where you can actually visualize this, and it'll be clear what's going on. So, well, there it is. So, so this is one of the functions which I think are it's just pretty powerful for the package. It's something called a discriminant space. And then for each facet, what we do is we try to find the axes that best separates the two cell types. So in the light color, so the discriminant space model is trained only on the reference data, right? So you have the light color and the dark color. The light color are the reference data sets. So if maybe I can zoom in here. So the light color is, like I said, the reference, and the dark is the query. So the discriminant space model is trained only on the reference. And then what we do is we project the query data onto that space. If things align well, then you would have the query fall pretty much onto where the reference data is, right? So now if you look, for example, at BM plasma versus myeloid, obviously there's a bunch of query myeloid cells that don't fall anywhere near where you should fall for the reference based on the reference data set. Yeah, and then if you look at, oops, sorry. Yep, and then if we look at CD4 versus CD8, it's a little hard to tell because there are so many cell types, but there's a bunch of query CD4 that fall onto where the reference CD8 is. So these are potentially where the misannotation occurred, right? Yeah, and then if you look at CD4 versus myeloid and CD8 versus myeloid, again, there's a bunch of strange things that seem to happen based on the annotation from single R. Okay, and now if we look at the good annotation, so this is if I just use the expert annotation, I don't use the single R annotation at all, then obviously we see things that align perfectly, right? So in the discriminant space where we try to find the axes that best separate the two cell types, things fall where they should. So that includes for CD4 versus CD8, which are transcriptomically not that far from each other. So yep. So this is just one example of the many functions implemented in the package. I think there's now over 25 functions in the package for different diagnostics, but this is just one of them where you can see the visualization of it. Okay. Yeah, so there's a lot of material for this workshop. So there's the BioC workshop repository with the link to all the materials, including the slides, the vignette, the Docker image, and a link to the Galaxy workshop, which I'll switch to right now. This package is going through a lot of reviews and iterations. It'll probably be submitted finally for publication into Bioconductor within the next week or two. So it should be available soon. But you can get the development version from the CCBHMS GitHub. Any questions before I switch to the Galaxy workshop? Yeah.
So you have that plot, which is okay. Can online people hear me? Okay.
I can hear you.
Yeah. So you have that. You have this plot showing like whether the annotation showing like if the single R and expert annotation match. But so the plot is nice, but so do you also implement like a more quantitative metric for,
absolutely, yes, yeah.
Okay, so like
many
see like a Concordex to promote my lab's work and
say again, sorry, I missed that.
A Concordex.
I don't use it right now in the package.
Okay, yeah, but so you do have some like quantitative metrics.
Yeah, where instead of getting a visualization, you get a number.
Okay.
Yeah. In fact, actually, so what you're seeing is actually, so every function in the package also has an S3 plot method. So what you're looking at is just the plotting of the output. But for all the discriminant space there, you can actually also get the data that produce this plot.
Okay.
So
awesome.
you get the discriminant vectors and the projections onto them, of the reference and the query. And you can also get things like the Mahanel-Bis distance from the query onto each reference data set. There's everything there.
Okay, awesome.
Yeah.
Thanks.
We have a question from the online audience. Cancer cell populations are usually not found in public references. Any recommendations on annotating cancer single cell data? Have you tried your package on cancer data?
I've tried it on many data sets. I don't believe I've tried it on a cancer data set, but that's certainly an avenue I'd like to explore. I'm also in the process of writing finalizing the vignettes. So I'm going to try to include many different examples. Obviously, I'll consult with Ludwig because I'm sure he has a couple of ideas in mind for that. So, yeah.
This is fantastic work, and I'm really excited to,
Yeah.
to use it in future work. But I'm curious, in your experience, if you have an idea of how often a published data set and its annotations are discordant with what you would have found from your reference based off this approach.
I have to confess that I've done it. Most of my tests have been specifically for development purposes, so I've been looking for data sets where things don't align. But I think it would take me a lot more trial and error to see how common that is.
Okay.
I suspect it's not that uncommon because I've seen data sets where you have up to 100 cell types. So I can imagine that there's probably a lot of weird things that can happen for a lot of applications. Yeah. Yep. I do have to go to the coding section soon, though. Yeah.
So like, what about, so like cell types have, they actually have a hierarchy. So like, would you in say future direction, like account for the hierarchy of cell types?
Yeah, absolutely. So there's obviously, so these are just statistical diagnostics, but obviously domain knowledge obviously should come into play when you do a diagnostic like this to see if things make sense, for sure. Yes.
Oh, cool.
Yeah. All right. So I'm going to switch to the coding portion now. For those who are online, Andrew Ghazi has very kindly agreed to help out if there's any questions for the code. The vignette is pretty massive, but as I said, I put as much as I can on there. So if I don't go through all of it today, you can still play with the code and try it. And please, feedback is more than welcome. So what I'm probably going to do here is zoom source, and then to go full screen here. Okay. Oh yeah, I should probably zoom in. Is 12 big enough? Or should it go bigger? That's good? Okay. 13? That might be too big. Oh, there's 13, compromise. Is that okay? Okay. Okay, there's a lot of text in the vignette, so, but mainly it's to kind of give more context as to what the functions do. Some of them go pretty deep into the implementations sometimes have a bit of advanced stats, so I won't go over them today, but hopefully we can eventually have a manuscript. I was playing around with the workshop last night, and some functions take quite a bit to compute, so please bear with me. So yeah, I'm just really loading here the scDiagnostics package, and then we'll do just a bit of adding some PCA reduced DIMs to some single cell experiment objects, and so we'll need to load scRNA and scatter as well. Okay, and then as part of the package, there are 2 data sets available. In fact, there's 3, but I'll only use 2 of them today. There's the reference data, which is the data that was used in the slides, the 70% curated reference data set. So the reference data set only contains the expert annotation. And then we have a query data set, which contains the expert annotation and the single R annotation as well. So we're going to load these 2 data sets. Okay. And then some functions are really designed to work on a data set that only contains 1 cell type. So we're going to subset our data and then just, for example, extract the data for just CD4 cells. So we'll do that. Okay. It's going to take a second here. In the meantime, I'll go to the discriminant space function. So this is the function that computes the discriminant vectors, does all the projections, and gives you a bunch of other diagnostics. So for most of the functions the arguments look very similar, except for the arguments that are specific to that function. So for example, there's typically a reference data set, which is a single cell experiment object. And then there's a query data set, which is again, also a single cell experiment object. And then typically what we do is we give the column, the call data column, for the annotation of the query and the annotation of the reference. And then there's a bunch of other arguments that are unique to that function. So that function also uses some random forest machinery in the back to identify important variables to distinguish cell types. So there's, for example, an argument called ntree, which is the number of trees used in the random forest. And okay, so that just finished. So now we can run this function. And then most functions have an S3 plot method as well, which allows you to basically just plot the object that's returned. But to Lambda's question, everything that is used to produce the plot is included in the return value of the function and much more. So if I just run this, for example, hopefully it doesn't take too long. It was pretty quick on my computer. Yeah, if you run it on your computer, it's going to be instant. So, yeah. Not sure why it's not producing. Oh, there we go. Yeah. So this is the plot that was shown in the slides, and as you can notice, things are not particularly well aligned based on the input. And again, if you notice here for the query, we actually used a single R annotation. And then just as an example, hopefully it doesn't take too long, if I switch from the single R annotation to the expert annotation, and then I rerun this. Oh, and this is just a box plot to see in the light color, the reference, and then the dark color, the query. If I run the code again, but with the expert annotation, you'll get the second picture we got in the slides, which is things were well aligned. So there we go. And like I mentioned, there's a lot of outputs given by each function, and there's a lot of arguments. Everything is, I would argue at this point, pretty well documented. So if you look at the help file for each function, you can check what each argument does and everything else. Just as an example here, what we can get is the Mahalanobis distance from the query to the reference data, and then the critical value based on the sample size as well. So just to give an example, this is one of the outputs you can get. So the critical value, for example, is 9.21. And then if any value falls above that value, it means that it falls pretty far off based on the Mahalanobis distance from the reference data. So I'm only printing the first 5 values, but I ran it with the expert annotation. If you run it with the single R annotation, you'll see a bunch of values that go above 20. So, yeah. Okay, that's just the first one. And then another thing we can do is, again, train a PCA model on the reference data and then project the query onto that space and see where things fall. And then, for example, this argument here, you can specify which PC combinations to plot. And you can specify the cell types. So for example, if I just plot PC 1s to 3, you can check again whether things align where they should based on the single R or expert annotation. If you want more PCs because you want to check, I think up to PC 10, there's quite a bit of variation that I explained in this data. So for example, if you want to print the first 5, you can adjust it to your liking. Okay. This is a box plot. So again, this is basically the principal component scores of the reference versus the query. And again, all these functions are based on the query being projected on the space of the reference. So we can see here, there's quite a bit of disconcordance between the query and the reference. If I switch, for example, to the expert just to show you what it would look like if you have a good annotation is things look much more well aligned. Okay, so yeah. I'm not going to run the MDS one because it's kind of slow, maybe at the end if I have time. But again, this is the same thing. You'll have the light color and the dark color for the reference and the query for each cell type. And hopefully things align well on top of each other. Now, for example, if we want to look at a specific cell type, for example, CD4, we can compare the cosine similarity between the principal component vectors if we run separately PC on the reference and PC on the query. So if things are well aligned, what we would get is kind of this perfect diagonal between the reference and the query. So here you can notice I'm using the expert annotation. So things look pretty good, well aligned, right? If I were to switch to the expert, you can do it, but you'd have to change how you subset the data at the beginning of the vignette. Then you would see that you wouldn't get this sort of nice behavior for this heat map. Okay. There's also canonical correlation, obviously, is very relevant for this type of exercise where you want to compare how the reference and the query align. So in canonical correlation, we're looking for separate directions in each data set that align well with each other. And obviously, if things align well, you would imagine that each of these vectors have a high cosine similarity and have a high correlation with each other. So here you can notice that the cosine similarity between the canonical correlation coefficient vectors, the y-axis starts at 0.85, right? So this is very good. Things align very well if we use the expert annotation for the query and the reference. Okay. If you were to switch to the single R annotation, you wouldn't get anywhere near what you're seeing now here. Okay. I can maybe go back later and do it, but I might just run out of time, so I'll just go on for now. I'll try to run this one. Hopefully it doesn't take too long. So this is one of the latest functions. Oh, it wasn't so bad. One of the latest functions that was added to scDiagnostics. So this is basically based on optimal transport theory. So this blue curve that you see here is if we keep sampling from the reference data set, and then we compute the cost to transform one data set to another, you would get this blue curve here, right? And then we repeat this process, but now we do it reference versus the query, right? So this blue curve is basically computed using the bootstrap, where we keep sampling from the reference and then computing the optimal minimum cost to transform one particular sample of the reference to another sample of the reference. And then obviously this is the threshold for a 5% confidence level, which again, you can adjust here. Here we can see that if we use the expert annotation, things align very well. I can show you maybe at the end if I have time, if I switch to the single R annotation here, this dashed line here is completely far off from the threshold. So, yeah. Okay. There's also functions to compute the overlap between the important genes to distinguish two cell types. So what this function here does is for each binary problem, so for example, CD4 versus CD8 or CD4 versus BN plasma, we compute the most important genes based on the reference to distinguish those two cell types, right? It uses the variable importance measure of random forest to do it. And then you repeat the same process for the query data sets. And then you can compute the overlap between those gene sets. So if I run this here, and the function uses a very fast implementation of random forest. So even if you have a lot of cells, it should go pretty quickly. So here we can see that so this is the overlap between the sets of important genes for the reference and the set of important genes for the query to distinguish the two cell types. Obviously, it doesn't look too good for many of them, right? A lot of them are below 0.5. If I were to switch, for example, to expert annotation, right, you can see that now things look much better. So nothing falls below 0.8. Okay. All right. Another very neat function here is called is calculate average pairwise correlation. There's many different ways you can use this function. You can, for example, change the correlation method. If you look at the documentation, you can change which PCs are used to compute these correlations between the cells. And in essence, you would, if you plot the output, you would get something that looks like this. All right. And again, ideally, what you would get is as high of a correlation in the diagonal, and the off-diagonal elements shouldn't be too high. So this is the output if you use the single R annotation, and then if I switch to the expert annotation, things will look much better. Yeah. So you can see in the previous heat map, there was quite a bit of correlation between the reference and query cells for query CD4 and reference CD8, which was the root of the issue for single R. So, all right. Yeah, there's a bunch of statistical tests that can be performed as well. I just put a few of them. If you go through the package documentation, there's way more. How am I doing on time? 7 minutes. Okay. Yeah, so this is basically a, if you'll go through the vignette, there's a lot more details, but this is essentially a statistical test with the boundaries at, depending on the level, the default is 2.5 on the right tail, 2.5 on the left tail. These curves look like they're the same, but they're not, because the Gaussian distribution that results from the null distribution depends on the sample size. So you can notice that the thresholds fall on different values. So, yeah. And then as you can see, for example, for CD8, it obviously falls outside of what you would expect. Okay. There's a permutation test for Hoteling's t-squared test. So Hoteling assumes that your data comes from a Gaussian distribution, obviously. So what I did is I implemented instead a permutation test to try to relax that assumption. And then we can see here that, for example, if we use the single R annotation, obviously we detect significant, we reject the null, right? Because we have a p-value of essentially 0 if we use the single R annotation, okay? All right, then there's another function here, regress PC. There's many different ways you can use this function. I only put one of them here, essentially what we do is, for this function, is we regress each principal component against the cell type. And then we have the proportion of the R-squared value for this regression problem, where we have one categorical predictor with, in this case, 4 categories. So we can check, for example, for the reference data, what's the R-squared value for PC1 when we regress PC1 against the cell type. Right? And then we can do the same thing for the query data set. And then we can also have the adjusted p-values for each principal component. And then this is the threshold. So if you fall above that threshold, so this is -10 log of the p-value. So if you're above that threshold, it indicates that that particular cell type is significant to explain some variation in PC1 or PC2, et cetera, et cetera. Okay? So we can see that beyond, say, PC 8 or 9, there's very little, the proportion of the data is not explained so much by the cell type. Okay. So there's also, as I mentioned before, a lot of the functionality of the package involves identifying cells that may deviate from its assigned cell type. So this detect anomaly function can be used in many different ways. So it can be used, for example, by if you have only a reference data set, and then it would identify outliers based on an isolation force just based on the reference. If you also have a query data set, the isolation force model is trained on the reference, and then we use the isolation force to predict anomaly scores for the query. So just as an example here, we have both a reference and a query. I'm going to use the single R annotation. And then there's a bunch of arguments you can use for detect anomaly. For example, the number of trees that are used for the isolation forest, what's the anomaly threshold that you can adjust. I put 0.6 based on my empirical experiments is usually a good value to use, but again, it's very data dependent. And then I'm going to plot the outputs. So what I'm going to plot is, well, let me first generate the plot. So we're going to plot the query data, and then specifically for CD4. So we're going to plot the query CD4 cells, and then there will be two colors, black and red. There we go. So the red cell types here are marked as anomalous based on the isolation forest that was trained on the reference, right? And then we can also plot it for the reference if you want. Yeah. Is there a question or? Okay. Yeah. Yeah. So yeah. So this is just one particular example that can be used. Any questions for the code or? Yeah. How much time do I have left? 2 minutes. Okay.
Hi, can I ask a brief question?
Functions I wanted to go through. I think maybe what I can do here is just go through one particular workflow that you might use to try to see what went wrong with the annotation. But this is just a very simple example. There's much more fancy things you can do with the functions that were implemented, but just for the sake of this workshop, what we'll do is there's a function called calculate cell distances. So this function will calculate the distance from every query cell to every reference cell. And then you can do some sort of analysis based on these distances. So let me run this first. And then I'm going to use the detect anomaly function to tell me the top 6 CD4 anomalies if I train an isolation forest model on the reference and then predict the anomaly scores on the query. So I'm going to store these. And again, I'm storing the top 6 anomalies for CD4 based on the single R annotation. Okay. And then what I'm going to do is plot the distance data for the query cells against the reference cells for the assigned cell types. And we'll see clearly that things don't look quite so well for CD4. Just give it a second. There we go. So, this blue curve here, these are the reference distances, the within reference cell distances, and then this red curve is the distance from the query to the reference for that particular cell. And then we can see that clearly there's very little overlap. And then if I do it instead for CD8, we can see that for some of them, things seem to line much more. And then finally, here, this is a function that computes something called the Hellinger distance, and another function that's called the Bhattacharya coefficient, and it's a measure of similarity between specific cells and the distribution of that cell type. So for example, for the Hellinger distance, the lower the value, the better. So for example, if we look at this particular cell, the Hellinger distance for this cell based on this versus the distribution of CD8 is very low. So that's a good indication. And then you can also look at the Bhattacharya coefficient. The higher the value, the better. Obviously, it looks like most of these cells might actually be CD8 based on these Bhattacharya coefficients. So I think I should stop here and take any questions. Thank you.
Yeah, I was just wondering, have you guys looked at alluvial plots as a way of comparing two labelings?
Say again, sorry, I missed the first one.
They're called alluvial plots.
Okay.
So it's like imagine sand flowing between two labelings.
Okay.
Like cluster 1 splits into cluster 2 through like 1, 3, 5, and,
Okay.
and the other clustering is like it's a
Okay.
flow type plot.
Okay.
I don't know. It's
Yeah.
might be a feature you guys might want.
Yeah, if you can
It's very
issue a,
simple.
yeah, absolutely, yeah,
It just shows up in papers sometimes. and
Absolutely, yeah. Thank you so much. Yeah.
Suppose you use so for like the cell type call, like you use like expert annotation for both query and ref. But so would your metrics and plots be used as a diagnostic for batch effect if I put like 2 different data sets in reference and query data?
Yeah, absolutely.
Yeah.
So for example, this regress PC function here, you can actually also include a query data set, and then you can regress on the batch, or you can regress on whatever you like. Yeah, absolutely, yeah.
We have another question. Reference data sets generally have different levels of granularity, resolution. Could you talk about designing a workflow to see what level of granularity produces the most variability?
Variability in what? Okay,
Well,
well,
which is best at,
so, okay,
or that's too fine, it's not getting enough.
right. Yeah, for sure, yeah, okay. It's definitely something I need to explore. I would imagine that, I can imagine the more cells you have. To be honest, it depends on the diagnostic. Yeah, I don't want to make a claim that I'm not sure of, but I would imagine that for certain diagnostics, you can get a much better confidence if you have more cells, but I would need to try it out, yeah.
It's been a long conference, and my brain's kind of fried. Yeah. Do you have to do this with principal components, or can you use whatever dimension reduction you want, and or have you tried that?
A lot of these functions rely on PCA. If you use other nonlinear dimensionality reduction functions like a VAE or like a kernel PCA, you can, but the problem with these methods is you're projecting the data on this higher dimensional space and you're kind of losing the information from the gene expressions. So I'm not for this.
Non-negative instead of nonlinear.
I'm sorry?
What about
Sorry.
non-negative instead of nonlinear?
I said non-negative? Sorry.
No, I'm asking if,
Oh.
because nonlinear, yeah, nonlinear has all its own problems.
Right, yeah.
Right, but non-negative actually has a lot of similarities to PCA.
Right. Oh, you mean non-negative matrix factorization?
Yeah.
Oh, yeah. Yeah, absolutely. So this is actually one I thought of possibly adding to the package, but I'm probably going to stop adding functions at this point and first get it on Bioconductor, and then we're going to add. Yeah.
Okay, cool. Thanks.
Nice talk.
Thanks.
So can you explain the utility of Wasserstein distance again?
Yeah, absolutely. So where is it? Yeah, so for basically the Wasserstein distances, so let's say we have a reference distribution and we have a query distribution, and these are high dimensional distributions, right? So optimal transport theory, basically we're looking for based on a cost function, and the cost function typically is the distance from one cell to another. But you can define your own cost function. What I ended up doing for this particular implementation is I did a weighted cost function where you put more weight on PC1 because it explains a higher proportion of variance. But whatever cost function you use, then what you do is you compute the cost of transforming this distribution to the other one. Right?
I'm going to have to jump in
Oh.
because we've got like 5 minutes to next, although I'm going to use my moderator privilege to ask my question. Are there any functions of these that you think that you would port back into the single R package to help improve either that or just spit out a undetermined or that sort of thing?
That would be nice. Yeah, sure. I haven't thought of it. I don't know if they would welcome it, but
Oh,
I would.
okay. All right, well, let's thank Anthony again. Wonderful. Looking forward to using the package. All right, we've got 5 minutes, then we've got final remarks.